Thank you very much, Dr. Flowers and Dr. Tomaso. You really set the bar high for me this afternoon. I'll try to live up to these expectations. But if you have any trouble hearing me, let me know and we'll try to adjust things. But welcome and good afternoon. We're going to focus on catfish genetic enhancement, but I'm going to use a couple examples that aren't catfish but are really relevant to the entire story. On the bottom left, that's our catfish genetics research facility here at Auburn. On your right is a in-pond raceway system, a commercial one in West Alabama. Of course, our objective is to use genetic tools to build a better catfish. And most of our focus has been on the farmer, but we also want to help the processor in addition to the consumers. Okay, some of the early scientists in our School of Fisheries at Auburn University, such as Ellis Prather on the left, and on the far right is a young version of Homer Swingle. They actually conducted the first catfish genetics research at Auburn, but they weren't aware of it because different species are different species because of genetic differences. So anytime that you do a species evaluation, you're actually doing genetic work. And they evaluated several different species of catfish and found that the channel catfish was the best one for fish farming. So then the first step in a genetic enhancement program, and that began at Auburn about 1969, is to look at different strains and determine which one is the best. And this is also important because the strain and the family have a big impact on how successful the other breeding programs are that we're going to discuss today. So after identifying the better strains, now you begin longer-term programs like selection, but we need to keep in mind that even though it's long-term, there's short-term benefits that are changing every generation. So here, this is the improvement that was seen in the body weight due to selection in the Kansas strain of channel catfish. And after 4 generations of selection, those fish are growing about 50% faster. In fish in general, if you have the patience to conduct selection for about 10 generations, in many species you're able to double the growth rate. So by the early 1980s, we were releasing genetically improved selected catfish to the industry. They were also, the farmers themselves, some breeding companies were also practicing the selection themselves. So by 2003, about 2/3 of the catfish industry was using selected catfish based on Auburn technology. So when you select for a trait, there's a chance that you might either positively or negatively influence another trait. So by selecting for body weight, there were slight improvements in reproductive performance, carcass yield, and disease resistance. But there was also a negative effect on tolerance of low oxygen. But fortunately, the catfish farmers are aerating their ponds every night anyway in order to make sure that the fish don't reach a stress point. So if we look at production in regards to pounds per acre, how many fish you're able to grow on a limited amount of water, so we're standardizing it. When we look at about 2003 or so, from 1980 to 2003, how many pounds per acre the farmers could grow had tripled. And part of that is due to improvement of culture and aeration, but also part of that's due to genetics. So also in the 1970s and 80s, Auburn began working on an interspecific hybrid between channel catfish females and blue catfish males. And these fish, depending upon the environment, grew 20 to 100% faster than the parents. Their feed conversion efficiencies, 20% better. They have improved disease resistance, better survival, better tolerance of low dissolved oxygen, higher harvestability, improved processing yield. So if we look at the overall performance and production, they're greatly improved, very valuable. And there was the results in commercial field verification trials were exactly the same as what we saw in small research ponds at Auburn. Here we have an example from West Alabama where you get almost a doubling in growth rate and about a 50% improvement in feed conversion efficiency. Nowadays, a large portion of our farmers are using very intense systems to grow catfish, such as split ponds, in-pond raceways, and traditional ponds with increased aeration. The parent species, the hybrid is essential for these systems to work. The parent species, channel catfish and blue catfish, they cannot tolerate this crowding. So we can't take advantage of this technology without use of the hybrid. So if we look at all these traits in combination, really this is the best example of genetic improvement in aquaculture ever. However, it doesn't matter if you have the best genes in the world. We can't help people or farmers if you can't produce commercial quantities of seed. And with this being two different species, there were mating blocks that made it difficult to make the hybrid. So it was more than 30 years after we had the first data indicating that it was a superior fish that there was any significant application on farm. We had to figure out how to overcome these barriers. So first, Auburn developed an artificial spawning technology using carp pituitary extract. This was commercialized by Gold Kiss, which later became Harvest Select. But there wasn't really a tremendous impact. You see, from 1997 to 2004, only about 1 to 2 million embryos were produced per year. So we continued improving the technology and switched to an LHRH spawning technology, and coupled that with some other improvements, and we were able to double to triple the efficiency, the output of the hybrid fry. So we used the LHRH and then protocols that we developed, and also borrowed the best ones from other places, and developed an LHRH technology with spawning bags, improved nutrition, better broodstock management, better hatchery procedures, and came up with a protocol that would work effectively commercially. Auburn partnered with Eagle Aquaculture to transfer this technology to the catfish industry. This is a picture of the Eagle team working on a farm in Mississippi producing hybrid embryos in a factory-type fashion. So Eagle came into operation in 2005, and they actually worked with the fish production part until 2010. So we see in 2004, 1.5 million hybrid fry. Eagle's very first year of existence, that jumped to 15.2 million, and by the time 5-6 years later, we're approaching 100 million hybrid fry produced per year. And now today, 2018, there were 275 million hybrid fry produced, which allows about 75% of the catfish farmers to use hybrids. So a large impact here. Then also, if we look at the number of the pounds per acre produced, how many catfish you can produce in 1 acre of water, you see about the time you start applying the hybrid catfish, the slope of that curve steepens considerably, and now we grow 5 times as many catfish per acre as what we did in 1980. But of course, our goal is to continue to improve. In this case, if we want to further improve the hybrid embryo output, again, strains are important. So by choosing the proper strain, we can double or triple the output of the fry, choosing the correct strain of female. And also males have an impact too. There's great strain variation for sperm production and also their ability to fertilize the eggs. Then after identifying those better strains, we can further improve the fry output by selection and increase the fry output by another 20 to 50%. We also want to use genetics to continue to make a better and better hybrid genetically from the standpoint of growth, disease resistance, and carcass yield. And again, there are significant strain effects. So in this particular example, if we take a Rio Grande blue catfish or an Alabama commercial strain of blue catfish, cross them with the same channel catfish female, the male is, the Rio Grande is letting those fingerlings, those progeny, grow 40% faster. So again, then what we want to do after identifying those better strains is we want to select for channel catfish and blue catfish that will produce an even better performing hybrid. In order to do this, we need to know combining ability. We have general combining ability of either the male or the female parent, and we also have specific combining ability, which is controlled by dominance genetics. So in this case, we're looking how individual pairs combine. So if we look at the pie diagram, at the blue color indicates that the female has a significant effect on the growth performance of the hybrid fingerlings. And if you look at the red pie, it shows that the blue catfish parent does as well. So what this tells us, if we want to improve the growth rate of the hybrid progeny in the split ponds, if we select for a faster growing channel catfish, that should result in faster growing hybrids. If we select for a faster growing blue catfish male, that should also help increase how fast our genetically improved hybrids will grow. The story's a little bit different here for dress out percentage. You'll notice that the green pie is larger than the blue ones and the red ones. This is telling us that the specific combining ability is more important in this case, and we'll make better progress if we search for pairs of males and females that produce better progeny for dress out and select for those parents. So if we go back to taking channel catfish out of the river in the 1940s, 50s, 60s, then domesticating them, then selecting them, and then making selected hybrids, those fish that we have today, we're able to get 10 to 20 times more production than when we first took the wild fish out of the river. We're using other techniques to try to further simplify and make better the hybrid embryo production. We're evaluating a process called xenogenesis, which is a method of reproduction in which the successive generations differ from each other. This is analogous to human surrogacy, but in this case, instead of the host hosting a baby, the host fish is hosting the sperm and the egg from another species. So it's similar, but not quite the same. So in this case, we want to sterilize channel catfish male, and we do that by making them triploid, and this triploid channel catfish male becomes the host. Then as a blastula or a fry, or perhaps even as an adult, we inject stem cells from blue catfish with the goal of making a channel catfish that is only capable of producing blue catfish sperm. And we'll finish that story in just a second. But this technology was first done with salmonids, where they were able to make xenogenic salmon, mate the male and female together, and the resulting progeny are 100% rainbow trout. Then here's another example from Japan, where they sterilized a male zebrafish, introduced stem cells from either silver danio, a goldfish, or on the far right, a mudloach. And those xenogenic zebrafish males produce sperm from silver danios or goldfish, or amazingly, from the mudloach. So in our case, the fish on the right are normal channel catfish. They are produced from the mating of a normal channel catfish female and male. The individuals on the left are hybrid progeny that were produced by mating two channel catfish. So a normal channel catfish female was mated with a triploid channel catfish male that's only producing blue catfish sperm, and you get 100% hybrids. We've had success. We were able to produce as many as 1,000 fry per female. That's actually a low number, so there are several students in the audience that are working on this procedure to make it more efficient so it can be commercially applied. Of course, we have made tremendous progress in the last 20 years in the area of genomics, switching to the molecular aspects of genetic improvement. And so now we can identify every DNA, every genetic difference in existence between two individuals. And a collaboration led by my colleague, John Liu, has made great progress in this area with catfish. In this particular case, this is what's called a genome-wide association study, and we're looking at a bacterial disease of catfish, ESC, and blue catfish are almost totally resistant. And this figure, the interpretation of this figure is the chromosome number 1. There are DNA markers that are locating the general area on the chromosome that is totally responsible for the ESC resistance in blue catfish. So we have markers that tell us what to look for. And channel catfish are much more resistant to columnaris than blue catfish. So this GWAS analysis is telling us there's 4 different chromosomes of channel catfish where the primary genes are located for disease resistance of columnaris for channel catfish. So this allows us to select, based on the DNA markers, the best genes from channel catfish and blue catfish and try to put it into the same fish to make a synthetic breed with enhanced disease resistance. And the preliminary results so far are real encouraging. The first data where we've done the preliminary marker-assisted selection, these fish showed very high disease resistance. So that's something that hopefully we can impact and help the farmers in the near future. These molecular findings and the early genomics work has allowed us to look at genetic engineering in fish and animals. Our lab was the first to produce a transgenic fish in America in 1987. It was the 4th lab in the world to do that. Most of the early work focused on growth hormone gene transfer, and other labs were able to develop tilapia that were growth hormone transgenic that grew 2 to 4 times faster than normal. The salmon researchers developed salmon that grew anywhere from 2 to 10 times faster than normal. In the early days, we always told people we're only enhancing the growth rate. The maximum size of the fish is only a little bit bigger. The important thing is they're growing faster. Well, the Koreans changed the story. On the bottom left, we have a very slow-growing species, mudloach. They're about 4 inches long, but they were able to make growth hormone transgenic mudloach, the bigger fish in the picture, that weigh 30 times greater than the normal maximum size. And so those fish are about 10 inches long versus the normal 10-inch fish. But we need to bear in mind, some people think mad scientists can do everything, but Mother Nature has rules and draws limits. We can only produce chickens that produce a certain amount of eggs per day, and we can't exceed that. We also there's limits on how big we can make a chicken egg. If it gets too big, the chicken isn't able to lay it. So Mother Nature imparts limitations. There's only so much we can do. In the case of growth hormone gene channel catfish, we're able to double growth rate. There are changes of the muscle ultrastructure, which might actually lead to better texture and make Dr. Flowers happy. These growth hormone transgenic catfish are able to tolerate temperatures slightly less, so their temperature tolerance is lowered. They can also tolerate slightly higher salinity concentrations, which isn't surprising because growth hormone has a major role in osmoregulation. We're also doing a lot of work looking at transgenic disease resistance. In this case, we're putting an antibacterial protein gene from the Cecropus moth into the channel catfish, and we were able to enhance disease resistance by about 2 to 4-fold. However, the results were more inconsistent than what we had hoped for. So some of my students are now looking at another antibacterial peptide from alligators. Alligators, they fight each other, they have severe wounds, but they don't get infections. And it's been discovered that they have an antibacterial peptide called cathelicidin. So here we have some catfish pathogens, and we're comparing antibacterial peptides from some aquatic organisms. The second row is where we've placed the alligator cathelicidin with the pathogens, and you can see that that's the Petri dish with the least growth of bacteria. So this area of research seems very promising. In this case, Rhoda compared the Cecropin bee from the moth, the cathelicidin from alligator, the cathelicidin from sea snake, pluricidin from flounder. These are all antibacterial peptides, but also compared their power to ampicillin, and use these to inhibit the growth of 5 major catfish pathogens. The cathelicidin from the alligator finished first in every comparison, except in the case of the Edwardsiella tarda, the ampicillin appears it might be slightly more effective. So the cathelicidin from the alligator is really promising because of its broad spectrum and power. So we're interested in disease resistance, obviously, to increase production, efficiency, and profits for the farmer. But also if we have healthier, fitter animals or fish, that's a positive from an animal welfare aspect. Of course, genetically engineered fish, by definition, are not organic, but if we can reduce or eliminate chemical or antibiotic use, that seems, in my opinion, to be just about as beneficial as organic culture as well. So, of course, the farmer is interested in production. The consumers don't have the same goals as a farmer. The consumers are more interested in price and especially healthy, nutritious food. So we've tried to do an increasing amount of our research geared toward helping the consumer. In this case, what we're doing is we're taking the desaturase gene from salmon and transferring it to channel catfish. Desaturase enzymes are the key enzymes in the fatty acid pathway that leads to the production of omega-3 fatty acids. And of course, omega-3 fatty acids are extremely important in human health. They're associated with brain and eye development, cancer resistance, cardiovascular health, and protein and lipid metabolism. Channel catfish are a very healthy product, but they're low in omega-3 fatty acids compared to salmon and some other species. So our goal is to increase the omega-3 fatty acid levels in channel catfish. The initial results are positive. We're getting a 20 to 30% relative increase in omega-3 fatty acids. Catfish with increased healthy ratio of omega-3 to omega-6 fatty acids. And also in this case, by inserting one gene to address the fatty acids, we're also enhancing some other characteristics. These desaturase transgenic catfish have increased growth, uniformity, and disease resistance. We're not doing research in this area, but it's an interesting type of genetic engineering. It's not all genetic engineering projects should be conducted. This one's probably not environmentally friendly. In fact, I would say it's not. Fish in the Arctic and the Antarctic can't survive with the high salt content. The water actually is below 0 degrees C. So a normal fish, their blood would freeze. But these Arctic species have antifreeze proteins in their blood. So some Canadian scientists took the antifreeze protein gene from winter flounder and they transferred it to salmon, trying to get them to grow closer to this Arctic circle. These salmon did produce antifreeze protein, but it wasn't a sufficient quantity. Their cold tolerance was unchanged. They also transferred these genes to goldfish, and again, the lower temperature tolerance did not change. But the survival of the goldfish at lower temperatures that you usually find them at, the winter mortality did improve. They had higher survival at cold temperatures. This is the first commercial application of transgenic fish. It's not for food. Many of you have probably already seen these. We have a normal zebrafish on the bottom left, and they have transferred the fluorescent protein genes from jellyfish into these fish. And these are sold in Auburn and all across the United States, different places in the world. So you have aquarium fish that glow in the dark. So recently, we have a new tool, gene editing. This is different from genetic engineering. In the case of genetic engineering, you are inserting DNA or genes from the outside. In the case of genetic gene editing, you're essentially developing a pair of DNA scissors. So you're introducing proteins and RNA that are temporary, and they are deleting, they are taking out DNA sequences which disables a gene. So the whole idea here is to disable a gene, but there's no introduction of any foreign DNA. So we've done this to try to increase growth rate. MC4R is a metabolism gene. We're able to remove about 200 base pairs. These mutants grew about 70% faster than normal, and their feed conversion efficiency was about 50% better than normal. These are natural mutants. there is a mutation of the myostatin gene. Myostatin is an important growth regulator in animals. But Belgian Blue and a couple of other cattle breeds have a natural mutation of the myostatin gene, which allows them to grow faster, and you can see the double muscling. And also the cow on the bottom left, you'll notice that she has a scar. One of the problems with this natural mutation is the fetal calves are growing too fast. So sometimes, fairly frequently, these cows have to have C-sections because the calves are too big to deliver. This natural mutation has also been found in a couple of human beings and a few dogs. Maybe somewhere out there are some future Auburn tigers that are myostatin mutants. So we induced this mutation in channel catfish using gene editing, and the mutant individuals grew about 30% faster than the controls, upwards to 30 days. The second row of pictures are histograms of the showing the number of muscle fibers. On the left is the control, on the right the mutant, and you see that the mutant has about 50% more muscle fibers than the control. Hopefully that'll have a positive impact on not only growth, but we're evaluating to see if it has positive or negative effects on texture. Okay, so there's been some very interesting genetic engineering work and gene editing work, but this has resulted in almost no, very little impact on fish farming and on food production. And the major reasons for that are it's a new technology, relatively new. So we have government regulation, we have groups that are opposed to genetic engineering technology. There's lawsuits, there's concerns about environmental risk, there's problems with public understanding of the technology and public education, and also concerns about food safety. The first of these issues I'd like to look at is environmental risk. There's been a considerable amount of research that shows in regards to fitness traits such as reproduction, the ability to catch food, the ability to avoid predators, reproduction, transgenic fish are less fit than wild fish and non-transgenic fish. If they were to accidentally escape, likely they would be eliminated because they can't outcompete the native fish. However, we have to be scientifically cautious, not rash, and do things the right way. So one of the things that our lab's been looking at is reproductive control of these fish. The goal is to have total reproductive control of the fish so that they cannot make any fry without help from the hatchery manager. If you're able to do that, you eliminate almost all environmental risk. So how do we do that? And this research is not only relevant to transgenics, but there are people who are concerned about fish farming and domestic fish in general, like the controversy we have in the salmon industry. There's concerns about exotic species that people want to apply for recreational fishing or fish farming. There's concern about the environmental impact of hybrids, both from a recreational standpoint and fish farming standpoint. And of course, with the new gene-edited fish, we need to worry about everything we can, so we add that to the list as well. So one approach is to actually use transgenics. So we've been looking at repressible transgenic sterilization. In this case, what we're trying to do is knock down the primordial germ cells. The primordial germ cells are the precursors to sperm and eggs. And the genes that control their migration during embryonic development to the genital ridge are, here's a couple examples, Nanos, dead end, and Vasa. So we developed some gene constructs using short hairpin RNAi or overexpression of cDNA to stop the migration of the PGCs. Then they die, and the resulting individuals cannot produce sperm or eggs. So here's on the top right, you see normal testicular development of a channel catfish. On the bottom, you see the normal ovarian development. And here are individuals with our sterilization genes inserted. In this case, these are females. They either don't have an ovary, or the ovary is totally empty and atrophied. Here are males, one case where there's no gonad at all. This was an unexpected result. We expected no gametes, but we didn't expect that they wouldn't develop any gonads at all in some cases. So, of course, if you had a farm with these fish, it wouldn't last very long because all your fish are sterile. So we have to have a way to reverse the process, in other words, to stop the expression of that sterilization gene so that we can have broodstock to produce our sterile production fish. In this case, we add copper to the water during embryonic development, and that stops the expression of the sterilization construct. So the top fish is an embryo that's developed in the water without the copper, and you can see the gonad is missing. In the bottom fish, it was incubated in water containing the copper, so the sterilization construct is turned off. It's still there, but it's turned off, and that allows the fish to develop gametes so that you have broodstock for your farm. An alternative approach is using the new gene editing technology, and in this case, we're trying to artificially mutate the key reproductive genes like luteinizing hormone, follicle stimulating hormone, and GnRH. We had success. On the left-hand side, you see eggs from a gene-edited fish, and the initial experiments, only about 1% of the eggs were fertile and could hatch. You see the heavy fungal growth because the eggs are dead. And again, we have to have a way to reverse this so we can spawn the fish. And in this case, we use normal spawning hormones. We introduce the reproductive hormones, or in other words, we have hormone therapy in it to temporarily restore fertility to the broodstock. And the eggs on the right are developing normally and will have high hatch rate. So, and again, in this case, we have total control because they're sterile unless we inject them with the reproductive hormones. So in both of these cases, the transgenic technology and the gene editing technology, we're very close to having this perfected. We still need to verify it with another generation. The other thing is we have to verify that these fish, we've got the growth enhancement, the disease resistance enhancement. We need to make sure the sterilization doesn't take that away. So the next issue, an interesting one, is public education. And I'm not sure exactly how to solve this one. A lot of the public, they're educated by this means, and this is how their opinion is formulated, with a little dose of misinformation from people who are against these genetic technologies. And to put things in perspective, one researcher in 1994, Thomas Hoban, surveyed people in North Carolina. Now, a high percentage of our fruits and vegetables are hybrids. So everyone in this room has eaten a hybrid fruit or vegetable. So the question in the survey was, have you ever eaten a hybrid fruit or vegetable? 60% of North Carolinians said no. And then the follow-up question was, do you think it's ethical to eat a hybrid fruit or vegetable? And 60% of North Carolinians said no. So we have a gap in the information knowledge. Now here's one that he did across several developed countries. And the question is, and it's easy, it's true or false. Ordinary tomatoes, so natural tomatoes, do not contain genes. Genetically modified tomatoes do have genes. Obviously, the answer is false. Only 45% of Americans could answer this question. And if you have French background, I apologize, but only 32% of the French could correctly answer this question. And even highly educated people can have problems understanding genetics, or they do not want to understand genetics. These are two of my daughters. The one on the right is Amy, and I'm going to tell you a quick story about her. When I wrote my first book, I gave a copy to all of my children. She was a teenager at the time, and apparently she didn't like freshman biology very much. So a couple months after I had given her the book, and she's a person with a dry wit about her. So I asked her, Amy, how did you enjoy my book? And she said, well, Dad, I opened up the book and the first word I saw was allele, so I closed the book. But this problem with biological technology transfer is not a new one. Vaccines were invented in the late 1700s, early 1800s. Most of us would agree that most vaccines are useful for human health. But this is an illustration from 1802 from the Anti-Vaccine Society trying to educate the public about the danger of the cowpox vaccine, showing a picture with cows growing out of people's heads, arms, rear ends, etc. So this public education and accepting of new technologies is not a new problem that society's dealt with. The issue of food safety. Now it is possible, really the only potential food safety issue with genetically engineered food is the potential for allergenicity. For instance, if you put a peanut gene into a catfish and a person with a peanut allergy ate the fish. However, the real food experts indicate even the possibility of that happening and eliciting such a response is very remote. It's highly improbable. So there's been reviews done by the U.S. National Academy of Sciences, the Royal Society of London, FAO, WHO. The conclusion of all was that there's no logical reason why transgenic meat should have any health hazards. In Europe, they have even stronger anti-genetic engineering sentiment than here. But even the European Food Safety Authority did a review, and despite that culture's negativism toward this technology, they concluded there's no logical reason that genetically engineered food poses a health risk. So how about the future? Well, the future is already starting to arrive. AquaBounty has commercialized transgenic salmon that reach market size about twice as fast as other genetically improved salmon. In 2015, for the first time, a government approved the consumption of transgenic fish. The FDA approved these fish for consumption in the U.S. However, no Americans have consumed these fish yet because it did not include being able to grow them here. The other problem was there's some U.S. laws. These fish are not labeled, so that triggered an import ban. So even though it's legal for us to eat them, it's illegal to introduce them to the country. But we'll finish that story in a second. So then next, Canada approved these fish, and growth hormone transgenic salmon have been sold in salmon and consumed by the public. Now there's been about 3 batches and about 15 tons of transgenic salmon sold and consumed in Canada. AquaBounty has an FDA-approved grow-out facility in Indiana, so they now have approval to grow them in America, eat them in America, but not bring them into the country in America because of this import ban. However, that is in the process of changing right now. In December of 2018, the USDA, well, let me back up for just a second. All of you have been eating transgenic food for a long time because the vast majority of our corn and soybeans are transgenic, as well as several other food items grown or consumed in the U.S. However, there is no labeling required, which of course upset the anti-genetic engineering people or anti-GMO people immensely. So partly because of this transgenic fish problem, the USDA in December of last year passed the first labeling laws. So starting in 2020, in your grocery store, you'll start seeing packaged food that says derived from bioengineering. And this will allow now that will lift the import ban, and transgenic salmon will actually be sold in the U.S. The people who are against this technology are less than pleased with the new law, even though for the first time in America, genetically engineered food will be labeled. However, it will be called bioengineered instead of the commonly used genetically modified. The other thing they don't like is the food packer has 3 choices. They can indicate the bioengineering using text. They can indicate bioengineering by the logo that you just saw, or they can do it with a digital link. And this really upsets the anti-genetic engineering people because they think people will be confused and who's going to go to the digital website to see what they just bought. So there's other things that aren't great about this law. They're defining bioengineered as foods that contain detectable genetic material that has been modified through lab techniques. Well, there's a lot of genetic modification in the lab that's not genetic engineering. So if they enforce this strictly, there could be other consequences. And the other thing that has some people upset is that the law only pertains to bioengineered food that contain genetic material. So oils, starches, sugars that come from a bioengineered organism but do not contain DNA, those don't have to be labeled. And the people who are against the process itself, of course, don't like this aspect of the new law as well. So the battle continues. Actually, in Africa, we have scientists now that are taking the public education problem to task and are trying to educate the people. They have organizations that educate the people on the benefits and the scientific-based benefits and risk of genetic engineering. But of course, the anti-GMO people, they continue to be active as well. Now, it forces me to finish with the fact that, well, what are we going to do about gene editing, the new genetic technology? So in this case, we're not adding, it's not genetic engineering. You're not adding any genetic material. What you're doing is artificial mutation to disable genes. So the FDA, they like to continue expanding their definition of drug. So it looks like they think that this technology comes under their purview. So it appears that gene-edited projects, a gene-edited fish would be regulated. Now, in the case of AquaBounty, they have a gene-edited tilapia. Argentina and several other countries have concluded that there's no need to regulate gene-edited animals. So AquaBounty will soon be selling gene-edited tilapia in Argentina, where the Argentinians approve. Now, part of the problem is some countries focus on the product. Is the product different than the natural product? That triggers regulation. In the US, there's just as much or more emphasis on the process. So the process, how you do it, triggers FDA to regulate. So we could have two fish, one in my left hand, one in the right hand. They are exactly the same. They have the same mutation. But one of them, the mutation was natural. The other one, the mutation is exactly the same, but it's artificial. The fish on the left is not regulated. The fish on the right, even though it's identical, will be regulated because of the process. Okay, so the future, getting back to the genetic enhancement. Most of these examples were using a single genetic enhancement program. There are examples where multiple ones have been used, and my prediction is in the future we'll be using catfish that are selected, genetically engineered, edited, and hybridized, all in one type of genetically improved fish, to take advantage, just like you use multiple tools to build the best house possible, we'll be using multiple genetic tools to build the best fish possible. And the research, that we have farmers that are concerned about all these international students sitting in the audience. But in reality, the international students and scientists that come here, they benefit us much more than what they take back to their home countries. The research, the Auburn-based research you saw here, was accomplished by students and scientists and staff that came from 27 different countries spread throughout the whole globe. And my last acknowledgement is I couldn't take time for each slide because you're probably getting tired of listening to me already anyway, but all of this research couldn't be accomplished without hardworking, intelligent, loyal, dedicated students that have been in our program over the years. So thank you very much. It's not the end, it's only the beginning. Let's make a bright catfish future.